Hi, I'm going to talk about the structure of DNA. So I'm going to talk about the repeating units of DNA, what they are, how the whole thing fits together. I'm going to talk a little bit about the bases of DNA and base pairing, and just start talking a little bit about transcription, but I'm going to leave most of that for the next video. Okay, so when we were talking about the biological macromolecules like protein, carbs, and lipids, we were talking about how monomers were the repeating single unit that made up these macromolecules. Well, with the exception of lipids, but we talked about how amino acids were the repeating unit of proteins and how simple sugars or glucose was the repeating unit of carbohydrate. So in the same way, the monomer of a DNA or even an RNA molecule, but I'm mostly talking about DNA right now, is the nucleotide. So what is the nucleotide? The nucleotide is this drawing here. So it has three components in it. I'm just going to hide this. Okay, so we've got this phosphate group here. That's what this circle's representing. We've got a sugar. The name of the sugar is deoxyribose. And those are going to be making the up and down parts of the ladder, the DNA ladder. And then we have a base. So the bases, there's four of them in DNA, and there's another one as well when we get talking about RNA, but I'll talk about that a little bit later. So the four bases in DNA are adenine, thymine, guanine, and cytosine. I just wrote in the letter there, and I'll talk a little bit more about each of those in a second. So I'll just move my face again here. So in this cloud here, this part that I blew up, I've shown you where that goes up here. So that's this. And so you can see how this repeating unit is actually making up the DNA molecule here. So we have the ladder, or the long parts of a ladder. They probably have a name, but I don't know what it is. And then the rungs of the ladder are these bases. So you can also see that the bases are differing sizes. You'll have like a larger one and a smaller one, a larger one and a smaller one. So you always have a larger one and a smaller one. These rungs on the DNA molecule are uniform size. They reach the same size. So you always have one of these smaller ones and one of these larger ones. Oh, there, myself there. Okay, so if we could zoom out, I just drew a really quick picture here of a DNA, like what you're probably used to seeing when you think about DNA, like this double helix. So now there's my base making up the rung of this ladder. I've got my phosphate and I've got my sugar here. And so the repeating units would just continue like that. So hopefully that makes sense. So I mentioned that there's a small molecule and a larger molecule, and here they are named. So we've got the larger molecule, and those ones are called purines, and the smaller molecule, and those are called pyrimidines. So the purines are the adenine and the guanine, and the pyrimidines are the thymine and the cytosine. If you want to come up with some mnemonic or some way to remember this, because really this is just memorization here, then that would probably be a good idea. So each rung on the DNA molecule is going to consist of a purine and a pyrimidine together. So, and they go together in a very specific way. So thymine always bonds to adenine, and guanine always bonds to cytosine. So you can see that on each rung of the DNA ladder, you are going to have like a uniform set. So it's kind of important to note too that they can only go together this way because of the way they bond together. So thymine and adenine form 2 bonds, and guanine and cytosine form 3 bonds in between them. And these are hydrogen bonds. They're quite weak bonds because they need to be easily broken apart as the DNA molecule unzips. So let's leave that thought there for a minute, just that whole unzipping concept, and think about these complementary base pairs. This is kind of a common question that you would often get on a biology test here, just to ask you if you knew the complementary base pairs for each of these. And so this isn't terribly difficult. You just have to remember that A's and T's go together and C's and G's go together. So on this side of the molecule, if you could just write down what base would go here. You could stop the video for a second and just work this out for yourself and then come back. That would probably be a really good idea. Okay, so hopefully you did that, and hopefully you didn't find it terribly hard. It really is just remembering that the A's and T's go together and the T's and A's go together. So A, the complement to that would be T, and T would be A, C would be G, and so forth. So those are the complementary base pairs on the strand of DNA. Okay, so what we want to discuss is how DNA, being the template or the blueprint, is what we call transcribed to RNA, and how that is translated into a protein. So we're going to probably look at most of that in my next video, but I do want to talk a little bit about RNA and the RNA molecule, which is another nucleic acid, so another biological macromolecule. So it's also a nucleic acid, like I just said. Instead of deoxyribose as a sugar, it just has ribose, and that's why it's RNA as opposed to DNA. An interesting thing is that it's only a single strand, whereas DNA is a double strand. So RNA is a single strand. And the other difference between RNA and DNA is, well, the DNA has the thymine as one of the bases, and RNA has a different base, which is called uracil. So if we were going to transcribe DNA into an RNA molecule here, we would be essentially doing the same thing as finding the complementary base pairs, but all we need to do is to make sure where we want to say T, we're going to say U instead. So here, the T is going to go with the A, and the A is going to be a U. The complementary RNA is going to be a U for uracil. G's still C's. T can still be transcribed to A, but again, A must be transcribed to U, to uracil, instead of thymine. So what we're making here is a RNA molecule. And the RNA molecule, well, it's a messenger RNA molecule, and it's a messenger because it's taking a little bit of coding out of the DNA, which is in the nucleus of the cell, and it's taking it out into the cytoplasm, where it's going to be read by the ribosomes, and it's going to actually be translated into amino acids, which, as you know, are the building blocks for protein. So from DNA to RNA is called transcription, or this is transcribing DNA into RNA. And the specific type of RNA it is because you're going to see that there's more than one type of RNA, but this is messenger RNA. So the M for mRNA stands for messenger. And that makes perfect sense because it's taking a message from DNA and it's taking it out into the cytoplasm and it's delivering that message, which is a code, which is going to tell the machinery in the ribosomes how to read it and make proteins. So what we've looked at so far is just what DNA is, how it's transcribed into messenger RNA, and then our next video we'll talk about how that gets translated into proteins. And we're just going to leave it there for today, and we'll talk about all of that in the next video.